So, the morphological changes that are usually seen in an ameloblast prior to deposition of enamel is that the cell is going to become tall columnar. The organelles are going to get aggregated to the distal extent or the distal part of the cell, and the nucleus is going to shift to the proximal end. So, the nucleus shows reversal of polarity with all the organelles coming onto the distal end of the ameloblast. The organic matrix formation primarily begins by deposition of enamel by the ameloblast. Now, there are two important things that are happening over here. One is the formation of the Tomes process. The Tomes process is actually a distal extension of the ameloblast into the newly formed dentine. So, remember that the dentine had to be formed first before the pre-ameloblast could get converted into the ameloblast. So, that is what is happening here. In the first layer of dentine, you have these ameloblasts that show these triangular processes. These are called as Tomes process. So, Tomes process is actually an extension of the ameloblast into the newly formed dentine. This Tomes process shows a third extension more, one more extension which is called as the distal extension of the Tomes process. So, now the Tomes process also has two parts. One is the proximal part of the Tomes process and one is the distal extension of the Tomes process. The proximal part of the Tomes process will form the interrod enamel. So, you have the proximal process of this ameloblast, the proximal process of that ameloblast together forming a canal. Inside that canal, you will have the distal process of the Tomes entering, and the distal process will secrete the rod enamel inside that canal which is formed by interrod enamel. So, the interrod enamel actually acts as a delineating membrane into which the distal process will go and deposit the rod enamel. Now, once the rod enamel starts getting deposited and the crystal starts growing, it squeezes this distal extension out of existence. And once the distal process of the Tomes is squeezed out, it leaves a very fine layer between the rod and the interrod enamel. And this is called as the rod sheath. So, now do you understand how you have the rod sheath, the interrod enamel and the rod enamel forming? Now, this enamel that has been deposited cannot be deposited as a 96% mineralized tissue. So, it is deposited only as a 30% mineralized tissue. The remaining mineralization and maturation of enamel happens at a later date. And this is called as maturation and mineralization of enamel. This is done by two important things, that is the smooth-ended ameloblast and the ruffle-ended ameloblast. Now, these smooth-ended ameloblasts primarily have the capacity to absorb all the water, and the ruffle-ended ameloblasts have the capacity to give in ions, the calcium ions. So, there is a modulation or changeover from the smooth-ended ameloblast into the ameloblast, ruffle-ended ameloblast in an 8-hourly process. So, every 8 hours, you will have the smooth-ended ameloblast getting converted into the ruffle-ended ameloblast, the ruffle-ended ameloblast secreting calcium ions for the next 8 hours, and then the ruffle-ended again getting converted into smooth-ended, taking back all the organic matrix. So, that is how the ameloblast will shift between the smooth-ended and the ruffle-ended ameloblast in order to make the enamel more mature. So, if I want to summarize the entire process of amelogenesis, remember it is these ameloblasts which are low columnar cells which gradually become tall columnar. There is organization of the organelles in the distal part of the cell, distal to the nucleus. The nucleus shifts to the proximal part. Remember why it is called as proximal part? Because from there, that is the area where it is actually supposed to secrete. But you have secretion happening from the distal end. So, all the organelles have come down to the distal end. Once the organelles have come down to the distal end shows an extension which we call as the Tomes process. So, the proximal part of the Tomes process will give rise to the interrod enamel. This Tomes process will have another distal extension, a further extension which will give rise to the rod enamel. the interrod enamel always will precede the rod enamel. So, you will have interrod enamel getting deposited first. Into this pit, you will have the Tomes process going in and depositing enamel. As the enamel crystal grows, the interrod enamel and the rod enamel will have a thin space between them because the Tomes process ceases to be out of existence. So, it gets squeezed out, and this small space gets filled up with organic debris, which we call as the rod sheath. Once the enamel has been deposited, the ameloblast shift between the smooth-ended and the ruffle-ended ameloblast till they can achieve a 96% mineralized tissue. So, that is what is amelogenesis. With this, we come to the end of a very important topic of amelogenesis.